So in the last section, we were talking about the endomembrane system. And you can see it here present in this cell. We've got a eukaryotic cell drawing, and we see the nucleus whose membrane is continuous with the endoplasmic reticulum. We see the Golgi body or Golgi apparatus here. We see the vesicles. We see the vacuoles. Here's the very large vacuole. And so the endomembrane system has its main job, which is to create, modify, package, and transport proteins. We know that proteins are what do all the work in a cell. They're the main workhorse of the cell. The main functioning part of a cell is the proteins. So that's going to be a really important role. However, we do have other jobs that need to be performed in a cell as well. So what we want to talk about here is what is the rest of a eukaryotic cell. Of course, we know that this means true nucleus, but we also want to know that eukaryotic cells are very complex. They actually have these compartments that allow for differentiation. Keeping in mind, the way that we get a compartment is through the use of a membrane. This is how we compartmentalize inside of the cell. We break the cell up into all these different little functioning parts. All of these compartments are usually called organelles. So we want to look at some of the other organelles that aren't necessarily involved in the production, package, and transport of proteins. That's what the endomembrane system does. We want to look at how do the other compartments help the cell survive and differentiate. So we also want to look at the idea of homeostasis. How do cells regulate their internal conditions? So the type of organelle and its function is going to help the cell have a specific job, but it's also going to help the cell control what's happening inside, especially if the environment is changing. So keeping in mind that homeostasis is what's known as dynamic. The environment is always changing and the cell has to be able to respond. When we talk about eukaryotic cells, we want to keep in mind we're talking about four main types: fungi, protists, and of course plants and animals. The interesting thing is that protists live in very, very diverse environments. So we actually see lots of different types of protists. So when we look at the eukaryotic cell, one of the big things that we're going to see is a vacuole. Vacuoles can have lots of different specific functions. In general, what a vacuole does is it provides a space for storage. So we can have a place to store food, waste, or water are the kind of three main things. So food, waste, and water. In protists, we actually see a very specific adaptation called a contractile vacuole. So when we talked about osmosis and water potential, and is water going to be moving in or out of a cell, we became aware that when something lives in water, we're going to be experiencing changes. Water coming in or water going out, and the cell has to be able to respond. One way that protists do that is through a contractile vacuole. So you were just doing an assignment with a paramecium, and that's exactly what the contractile vacuole does, is it helps regulate water, H2O, regulate how much water there is by contracting and pushing out that water. There are little proteins that surround it. So vacuoles are going to be really important for each cell being able to store specific things. One of the other things that we see in plant vacuoles is what's called the central vacuole. And this is going to be there to store water. It's very large in plant cells. And this is actually what creates turgor pressure, is the relationship between the vacuole and actually the cell wall. That's what creates turgor pressure. So together with the cell wall, the central vacuole and the cell wall are going to push against each other to create that pressure. And so the central vacuole in plants is usually sometimes even larger than the nucleus because it's containing all of this water. Another type of vacuole that we see are sometimes called zones. So we want to talk about two other types of vacuoles. There are lysosomes and peroxisomes. And we'll get to those, I think they're down here a little bit more later. But so a vacuole is just a membrane, basically a membrane sac, and they're also sometimes called vesicles, which we've seen that word already. So small membranes that compartmentalize the cell so that we can store certain things. An interesting thing is if you look at organisms who have poisons or toxins, like the brown recluse spider or the black widow spider, and we think about, well, how do they have this toxin in them and yet they don't die? They actually usually keep the toxin separated, the two parts of the toxin separated in different vacuoles, and when they release them, then the two parts of the toxin or the poison come together and are dangerous. So vacuoles are really important for helping the cell compartmentalize things separately. The other thing that we want to look at, and we will be talking about this much more later in the year, are these two double membrane-bound organelles, the chloroplast and the mitochondria. So all eukaryotic cells have vacuoles. All eukaryotic cells have mitochondria. Only plants and some protists have the chloroplast. So what we want to do right now is just get a very general overview of what the mitochondria and the chloroplast do, because we'll be talking about them a lot more later. What we notice is, again, they are compartments. We've got membranes, so these two are the double membrane-bound organelles, and these membranes create compartments. The mitochondria is creating compartments for the breakdown of glucose, which is C6H12O6. And so this is where respiration is going to happen. The big thing that these two have in common is a really high surface area. So an increase in the surface area. The more surface, the more reactions we can do. So these are kind of the two places where a lot of reactions, chemical reactions, are going on. Here we're breaking down sugars to get energy. And ATP is our energy source. So the mitochondria, lots of folds of the membrane. It might be hard to see on this diagram, so I'll draw it again here. We've got this double membrane that is going to actually fold in on itself. And so these folds we'll learn are called the cristae, and we'll talk about them a lot more later in the next unit when we get to cellular respiration and photosynthesis. For now, we want to know they're in all eukaryotic cells. They have this folded inner membrane, so there's lots of surface area for these chemical reactions to occur. When we look at the chloroplast, it's actually similar. Now keep in mind, when we talked about the endosymbiosis theory, these very well once could have been their own free-living bacteria that are now in a permanent relationship with the eukaryotic cell. Double membrane bound, have their own source of DNA, divide on their own, have their own enzymes. They have a lot in common, but their jobs are very different. The mitochondria's job is to break down glucose, to take the energy in these bonds and put it into a different molecule, ATP. Plants and protists, or what we know as autotrophs, they're going to actually be capturing solar energy isn't responding very well this morning. Capture solar energy to store store in chemical bonds. Store in chemical bonds. So photosynthesis is actually the building of glucose. So we have two organelles that have different functions but a similar structure because both of these are based on chemical reactions. So using the membrane to create, even within the organelle, its own little compartments. We're going to see that we need to create hydrogen ion gradients, and we need to transport hydrogen ions, and we need to capture this solar energy by exciting electrons. There's a lot that's going to go on in these reactions, and so we need a lot of surface area and a lot of different compartments. So the main idea is that eukaryotic cells use membranes to have an increased surface area and to create these compartments. And now my cell can do different things. We can regulate what's going on in the cell. We can do more respiration or less respiration. And what that actually also has to do with is the number of those organelles. In this average picture of a plant cell, it just shows us one mitochondria. But certain plant cells are going to have hundreds, certain might not have any. If we look at the plant cell, we think that every plant cell would have a chloroplast. But if the chloroplast needs solar energy, what about the root cells? They're not receiving any sunlight. So it turns out that the roots, if you've ever looked at the roots, they're white, they're not green, because they don't have any chloroplasts. So we see this big idea of differentiation has to do with the number and the type of organelle that's in there is directly related to the function of that cell. So again, the number and the type and the use and the size, these organelle structures are related to the function of the cell. So structure and function, again and again we see that. So just real quickly, we want to talk about some other, these vesicles, the lysosomes and the peroxisomes again. So when you scroll down on the page, there we go. I'm going to try and get it to catch up. I think it's a few minutes behind. When we scroll down on the page, what we're going to see is that we have some different types of organelles that are going to help perform these two jobs of breaking down things in a contained area. So what we see is here we have our lysosomes, and so what we want to know about lysosomes is that this is a vacuole that contains digestive enzymes. So a lysosome is a vacuole that contains digestive enzymes. Not sure that my pen is going to cooperate today, so I'm just going to have to say it. A lysosome is a vacuole that contains digestive enzymes. And this is actually going to be really important for something that we call apoptosis, and that's A-P-O-P-T-O-S-I-S. Apoptosis is actually when the lysosomes are released and the cell self-digests from the inside out. And this is actually how the tadpole loses its tail, is these cells commit cell suicide, actually trigger cell suicide, so that we can control where cells are developing. And so we have lysosomes contain digestive enzymes, and then the other one we want to mention is peroxisome that breaks down hydrogen peroxide. It's another membrane vesicle that contains enzymes, and that will break down hydrogen peroxide. And that's where we'll stop for today.